Hello, everyone. Today we're continuing our deep dive of Richard Dawkins and Yan Wang's book, The Ancestor's Tale. In this episode, we're going to discuss the diversity of ecdysozoans and the topsy-turvy behavior of brine shrimp. So, let's jump right in. In the previous episode, we met the various members of the giant protostome clade Spiralia. Today, we're meeting the other protostome clade, Ecdysozoa, which includes Priapulida, Caenorhyncha, Loricifera, Nematoda, Nematomorpha, Tardigrada, Onychophora, and Arthropoda. All members of this clade are defined by their ability to periodically shed their exoskeletal cuticle as they grow, a process called ecdysis. This process has been extensively studied in arthropods, including even some fossil members like trilobites and Fuchsianhuiids. However, the molting process of other ecdysozoans has been generally less well characterized. It broadly involves detaching the former cuticle from the underlying epidermis, splitting along weak points, and then the new exoskeleton hardens. The earliest ecdysozoan molt is known from the Cambrian period, 535 million years ago. Based on the shape, it belonged to a member of either Priapulida, Caenorhyncha, or Loricifera, collectively called the Scelidophorans. A few Scelidophoran species are known from the early Cambrian, including Eopriapulides, Eocaenorhynchus, Quinscolex, and Schanscolex, so the molt could belong to any of these, or an unknown species. Given that all ecdysozoans molt, hence the name, and the oldest confirmed ecdysozoan fossils date to the very start of the Cambrian, that means molting most likely evolved in the Ediacaran. Also, since all ecdysozoans except the tardigrades, onychophorans, and arthropods are legless, wormy animals that live in substrate, that was probably the ancestral ecology for ecdysozoans. Sand is coarse, so an adaptation to living in sand would likely include developing a progressively tougher exoskeleton. However, a thick exoskeleton would preclude continuous growth, so as the exoskeleton got thicker, ecdysozoans had to split their growth into stages. This might seem cumbersome, after all, the ecdysozoan has only a short window of time following a molt to grow. However, a benefit of this system is that damaged tissues or missing limbs can be regenerated so long as molting occurs. Once the animal finishes molting, though, limb regeneration can no longer occur. So, what of the earliest record of ecdysozoans? There is one potential relative of tardigrades, onychophorans, and arthropods, known as Eulingia, that dates to the latest Ediacaran, 551 to 541 million years ago. Eulingia possesses bilateral symmetry and trilobate segments that are similar to arthropod appendages or annelid parapodia. Unfortunately, the uncertainty in its phylogenetic position among protostomes means we can't draw a lot of conclusions about it in the greater context of bilaterian evolution. Once we cross into Ecdysozoa, we have the same problem with respect to a 1.3 millimeter fossil known from China known as Saccharidus. Saccharidus dates to 540 million years ago at the very start of the Cambrian and is a globular sac-like animal covered with spines. Some of the spines were broken off in the original specimens, leading the researchers to mistakenly conclude that Saccharidus had pharyngeal arches and making it a deuterostome. It has a mouth but no anus, meaning any undigested material was expelled back through the mouth. This fact doesn't bear on our discussion in the ragworm's tail because the loss of the anus appears to be secondarily derived, not ancestral. Many Saccharidus specimens are preserved as deformed integuments with no internal anatomy, which is consistent with being a molt instead of the whole body. Therefore, Saccharidus is an ecdysozoan, but whether it is a primitive member of a living clade or sister to all extant ecdysozoans is unknown. Now we turn to the diversity of living ecdysozoan clades. First, Priapulida, also known as the penis worms, likely appeared in the Cambrian. Priapulids are unsegmented worm-like animals who possess an extendable mouthpart covered in spines, called a proboscis or introvert, and tend to live in the mud of shallow waters. While originally classified based on morphology with other unsegmented protostomes, genetics has rooted them among the ecdysozoans. Interestingly, a number of Cambrian trace fossils bear striking resemblance to priapulid burrows, such as Treptichnus pedum, which is the trace fossil that defines the start of the Cambrian. So it seems probable that they evolved around this time. This would make priapulids important early substrate burrowers, contributing to bioturbation in cyanobacterial mats. Aside from trace fossils, there are priapulid body fossils in the Cambrian, such as Schauheikingella and Paratubulochus from the early Cambrian Chengxing Formation. Some Cambrian priapulid-like worms are known, such as Autoya from the Burgess Shale, but these evidently fall outside of Priapulida proper, being in the paraphyletic Archaeopriapulida. Further out still, researchers demonstrated that the priapulid proboscis is homologous to the mouth parts of stem onychophorans, Hallucigenia, and Gentianopodia, and the stem arthropod Pamdilurion, implying that such a feature is primitive in all ecdysozoans. A group that has had a contentious history related to priapulids is the Paleoscolechids, who existed from the early Cambrian to the late Silurian. While they look overall similar to priapulids, some Paleoscolechids have characteristics that make them more similar to tardigrades, onychophorans, and arthropods. For example, the Paleoscolechid family, Cricocosmiidae, is similar to other Paleoscolechids in general structure, but it also possesses larger, serially repeated sets of trunk sclerites, bearing a resemblance to trunk sclerites of some primitive arthropod relatives like Microdictyon. For this reason, some researchers have argued that Cricocosmiidae could represent a grade of ecdysozoans transitional between the more worm-like priapulids and the primitive arthropod relatives. Whether the Cricocosmiid features are convergent or homologous has been debated. A 2022 paper found Cricocosmiids as stem priapulids, but a response to that paper argued that when certain characters are scored as uncertain instead of inapplicable, Cricocosmiids were found to be more closely related to primitive arthropod relatives than to priapulids. So, as of this writing, Cricocosmiids do appear to be a transitional stage between the more wormy priapulids and the worms with legs that gave rise to tardigrades, onychophorans, and arthropods. Another enigmatic group is called Cambrochlavida, which are spine-shaped sclerites known from the early to Middle Cambrian. Some researchers have dubbed them as belonging to ecdysozoan worms, but without more complete specimens, this assignment is dubious. That brings us to a fossil originally identified in 1993 named Protomelysion. Protomelysion hails from early Middle Cambrian rock and was originally described as having morphology reminiscent of bryozoans, but being far too thin. Then in 2021, Protomelysion was reanalyzed and assigned to stem bryozoa, meaning that bryozoans originated in the Cambrian, like most bilaterian phyla. However, a 2023 study found more Protomelysion-like fossils and argued based on them that the supposed animal is in fact a daisy-cladalian alga. Later that same year, another study analyzed the microphotographs of Protomelysion from the earlier 2023 paper and concluded that what was originally thought to be zooids were broken spine-shaped sclerites, putting the genus in Cambrochlavida. The paper cautiously assigned Cambrochlavida to crown Eumetazoa, but declined to specify further. Previously considered as sister to Priapulida is the phylum Caenorhyncha, the mud dragons. These are small, segmented, mud-dwelling animals who have spines that help them move in the sediment. As they are only a few millimeters in length, it makes sense that they would have few fossils. However, the discovery of the Cambrian Qingzhang Lagerstätte in China in 2019 revealed 3 new Caenorhynch species. Ctenophores, sponges, medusozoans, priapulids, brachiopods, lobopodians, arthropods, new chordates, bizarre 4-fold radially symmetric algae, and other organisms have been discovered at the site. The environment of the Qingzhang appears to have been slightly different from that of the contemporary Chengxing Formation, giving researchers an insight on how Cambrian organisms vary with the environments. Additionally, one stem Caenorhynch has been discovered, Eocaenorhynchus, dating to the early Cambrian, 535 million years ago. And previously considered as sister to these two groups is Loricifera, a phylum of microscopic substrate living animals. These curious animals evaded description until 1983, making them one of the more recently named phyla. One loriciferan fossil was discovered just a few years ago, Eolorica, dating to the Cambrian. This fossil indicates that loriciferans are possibly descended from a preapulid-like ancestor, but then later miniaturized from larger loriciferans. Those ancestors, though, would have been millimeters or centimeters in length. I said previously considered because Ecdysozoa was thought to be split into three major clades. One group containing Preapulida, Caenorhyncha, and Loricifera, one containing Nematoda and Nematomorpha, and one group containing Tardigrada, Onychophora, and Arthropoda. A fairly recent genomic study has argued that the first group is instead paraphyletic. The study also posited that Preapulida is the most basally derived ecdysozoan, and Loricifera is sister to Nematoda and Nematomorpha. If true, then that would explain the difficulty in phylogenetically placing the Paleoscolecids, because their body shape could possibly be ancestral for all three groups of extant ecdysozoans. Nematoda, the roundworms, are a highly abundant and diverse phylum of ecdysozoans. Many authors over the years have remarked on their ubiquity, as over 12,000 species have already been named. However, some researchers have estimated that there may be many times more in existence. Nematodes inhabit marine and terrestrial environments from arid deserts to freezing polar regions and can be free-living or parasitic. For example, the aptly named Cryonema lives in Arctic ice, preying on other nematodes, while Xalophora consumes nematodes living in the intestines of millipedes. Meanwhile, plant parasitism has independently evolved among nematodes at least 3 times, and animal parasitism has evolved at least 6 times. The most famous nematode, though, is clearly Caenorhabditis elegans. This worm has had its genome sequenced, revealing some 19,000 genes, and making it the first multicellular organism to have this done, after the unicellular E. coli and Saccharomyces cerevisiae. Humans, by contrast, have around 25,000 genes. C. elegans is also a model organism due to having a particular number of cells, called utelli, a feature which allows researchers to map out the cell fates through the animal's ontogeny. As for nematode fossils, there are no body fossils dating all the way back to the Cambrian, but it is probable that the phylum split from the others around that time. The earliest potential nematode fossils date to the early Carboniferous, about 345 million years ago, but strong doubt has been cast over this assignment. But a well-established nematode fossil, Eophasma, is known from the Jurassic Osteno-Lagerstätte of Italy, 200 to 190 million years ago, a region that was once an anoxic seafloor. The nematode was found alongside brachiopods, ammonoids such as the index fossil genus Sulciferides, brittle stars, acorn worms, coelacanths, sharks, and chimeras. Later in the Cretaceous, some nematodes have been found preserved in amber, being preyed upon by fungi. If you are shocked by this, then allow me to quote the paper. Quote, "Today this ecological niche is occupied by more than 200 species of the zygomycetes and imperfect stages of the ascomycetes and the basidiomycetes." Close quote. Sister to Nematoda is Nematomorpha, the horsehair worms. These worms are often parasitoids of arthropods. Bizarrely, when they possess a host, they force the arthropod to drown itself to release the worm. If Darwin was troubled by the theodicy of ichneummonid wasps, then we can only imagine how he would have reacted to nematomorphs. As far as fossils go, a nematomorph called Paleochordotes has been found in Dominican amber, 45 to 15 million years old, parasitizing a cockroach. And another, Cretochordotes, is recorded in early Cretaceous amber, 110 to 100 million years old, along with a nematode in the process of exiting its host. Next, we move on to Panarthropoda, which splits into the phyla Tardigrada, or water bears, Onychophora, or velvet worms, and Arthropoda. The exact phylogeny of Panarthropoda has been controversial, and studies have found some measure of support for every possible topology. The two most likely hypotheses are called Antennapoda and Tactopoda. The former states that arthropods and onychophorans are sisters to the exclusion of tardigrades, and the latter states that arthropods and tardigrades are sisters to the exclusion of onychophorans. Morphological and fossil data tend to support Tactopoda, while genetic data has found support for Antennapoda. Either way, panarthropods are descended from a paraphyletic group of animals called the lobopods, or walking worms. Some lobopods are ancestral to velvet worms, some to tardigrades, some to arthropods, and some to all panarthropods. Essentially, lobopods are tubular worm-like animals with unsegmented legs bearing claws. Some taxa, like Achaea and Onychodictyon, have been difficult to place because of how primitive their characters are, indicating that something like them was ancestral to all panarthropods. We've already done a video on tardigrades, which is linked in the description, and velvet worms will get a tail all their own, so let's stick with the arthropods. Some lobopodians share more features in common with the arthropods, such as Siberion, Carygmachela, and Pamdelurion. Slightly closer to the arthropods, we find Opabinia, a bizarre animal with a long proboscis and fan-like lobes running along its sides. Disagreements have come up regarding whether or not this creature had lobopod limbs, but more recent studies have confirmed that it does. True arthropod limbs appear to have resulted from the fusion of lobopodian limbs with the lateral lobes. Getting closer to crown arthropods is the stem-clade Radiodonta, which were among the first apex predators, containing such forms as the monstrous Anomalocaris. This was a major predator during the Cambrian, reaching up to 3.3 feet in length, which was far larger than most of the animals it lived alongside. Its large size and curled spiny mouth parts have long earned it the designation of top predator, but this ecology has been questioned. Evidently, the Anomalocaris spines are too fragile to have been used for attacking the mineralized exoskeleton of trilobites. Therefore, the predator more likely used them for hunting soft-bodied prey like Pikaia or Autoya. If it did attack trilobites, then it probably would have focused on those that had recently molted. Interestingly, trilobites with calluses tend to have it on the right posterior region. One paper notes, quote, "This indicates that either predators attempting to eat trilobites had a preference to attack from a particular direction, or trilobites had developed lateralized defensive countermeasures to prevent damage to more sensitive regions of the body," close quote. Although most early members were predators, some radiodonts opted to become gentle filter feeders, such as the late-surviving Agyrocassus from the Ordovician. Now we come to several groups whose phylogenetic statuses are debated. Arguments over homology versus analogy regarding certain fossil characters have left their exact placement contentious, so they may be advanced stem arthropods or basally derived crown arthropods. For example, there is a group of bivalved arthropods called Hymenocarina, such as Nereocaris and Canidaspis. This group shows a few transitional characters in the evolution of stem arthropods, such as the jointing of the limbs to become biramus, jointing of the exoskeleton, and specialization of cephalic limbs. Another group of potentially stem arthropods is the bivalved Isoxeida. Members of Isoxeida represent the earliest body fossils of arthropods, being part of the early Cambrian small shelly fauna. Next, Fuchsianhuida, including members like Shengzhengacaris, displays a mixture of primitive and derived characters. For example, they have specialized appendages behind their antennae, which can be used to sweep food into the mouth, indicating that this feature is primitive in crown arthropods. And megachirons, named for their large frontal appendages like Leonchoilia, are another possibly paraphyletic group close to crown arthropods, a place earned by the similarities in their limbs with crown arthropods. A 2022 paper, however, put Megachira, Fuchsianhuida, and Hymenocarina within crown arthropoda, so clearly the debate isn't over. We need to discuss one more extinct clade of possible crown arthropods before getting to the extant members, trilobites and their kin. Trilobites are probably the most easily recognizable of all Paleozoic denizens, containing some 20,000 species. Their body plan is the iconic cephalon, thorax, and pygidium, which looks superficially similar to a number of other Cambrian animals. Thus, trilobites were shoehorned by early workers into a clade called Trilobitoidea, along with several animals that they were not closely related to, such as Morella, Leonchoilia, and Opabinia. Later researchers, of course, drastically changed the position of trilobites. One common hypothesis is that trilobites are sister to chelicerates. More on them later. Despite their common body plan, trilobites are quite diverse. Some are over a foot long, while others are less than a millimeter. Some had large spines, while others were smooth. Some had large compound eyes, the lenses of which were made of the mineral calcite. Yes, they basically had rock-hard crystals for lenses. In contrast, other trilobites were deep-sea dwelling species and were totally blind. A number of species, like Protolenus, have been used as index fossils. Due to their hard exoskeletons, some trilobite lineages are excellently preserved, such as that of Acatoparadoxides, displaying their evolutionary histories. Their adaptability allowed them to last from near the start of the Cambrian to the end Permian, some 250 million years, longer than the non-avian dinosaurs. According to the book A New History of Life, the radical new discoveries about the origins and evolution of life on Earth, quote, some of the earliest occurring of all trilobites, the Olenellids, underwent total extinction at the end of the Cambrian, and in fact the entire nature of the trilobite faunas changed. Trilobites of the Cambrian had many segments, primitive eyes, and no obvious defensive adaptations on the body, such as anti-predatory spines, and were unable to do what modern-day pillbugs do when threatened, roll up into a tight ball. After the extinctions, and thus in the earliest times of the Ordovician period, newly evolving waves of trilobites had changed their entire body plan. Now virtually all reduced the number of segments. Many segments are easier for predators to break through during a predatory attack than fewer, thicker body segments, and had better eyes, defensive armor, and especially the ability to roll up into pillbug-like balls. Close quote. From the Silurian to Carboniferous, trilobite disparity declined, while at the same time, interestingly, crustacean disparity increased. Whether or not this inverse relationship was due to competition between the clades is unknown. However, the extinction event that wiped trilobites out at the Permian-Triassic boundary decimated some 9 in 10 of all species. Related to the trilobites is another extinct clade called Vesissicaudata that contains, among others, Sydneia, Emeraldella, and the clade Aglaspidida. Interestingly, trilobites and their close cousins radiated in the early Cambrian, while Vesissicaudates radiated in the late Cambrian. Finally, we reach crown arthropoda. This phylum is defined by having jointed, segmented limbs and an exoskeleton that consists of chitin. The earliest well-established record of this phylum dates to the very start of the Cambrian. The trace fossil is Rusophycus, which evidently represents trilobite activity, and Diplycnides. This phylum is found to bifurcate into 2 clades, Chelicerata, and, except for a few extinct forms like Marella, Mandibulata, including myriapods, crustaceans, and hexapods. Molecular clock studies routinely place the divergence of Chelicerata and Mandibulata in the late Ediacaran period. We'll go to Chelicerata first. The subphylum Chelicerata is defined by having chelicerae, highly variable appendages associated with the mouth. Evidently, chelicerae are antennual structures that were exapted for a new purpose: feeding. This has been gleaned from fossils hailing from the Middle Cambrian, 508 million years ago, such as Mollisonia, an early chelicerate, though with its chelicerae positioned very anteriorly on the head. Extant members of this subphylum include Pycnogonida, Xiphosaura, Eurypterida, and Arachnida. The first, Pycnogonida, the sea spiders, are a group of exclusively marine arthropods who live in environments from intertidal zones to the abyssal depths. They're an odd-looking group, having an elongate proboscis and a highly reduced abdomen, and they predominantly feed either by scavenging or preying on small animals. Due to their rarity and the lack of large genetic sampling, phylogenetic analyses of the class are still ongoing. They do in fact have a fossil record with members such as Cambropycnogon from the late Cambrian, 500 million years ago, Haliestes from the late Silurian, 425 million years ago, and Paleoisopus from the late Devonian, 400 million years ago. Second, Xiphosaura contains the horseshoe crabs, long regarded as living fossils because of their resemblance to fossils found well over 100 million years ago. As it happens, though, there is a fossil record of Xiphosaurans showing their evolution from more trilobite-like ancestors. Over time, the head shield enlarged while the body segments fused. This can be observed from Ordovician Lunataspis to Devonian Bellinurus, to Permian Paleolimulus, to Jurassic Mesolimulus, to the modern Limulus. Like the sea spiders, horseshoe crabs are marine and feed on small animals. And despite their long heritage, they have very few species. Today, there are 3 genera, 4 species in total, and even at the height of their diversity during the Carboniferous, there were only a few dozen species. The order greatly declined after the Permian Triassic extinction, leaving just the modern family Limulidae. The split between Limulus and the other 2 genera, Carcinoscorpius and Tachyplus, occurred in the early Cretaceous, while ancestors of the other 2 genera split during the Paleogene. Third, Eurypterida is another well-known order of chelicerates, the sea scorpions. Though they outwardly look like scorpions, the similarity is merely convergent. Like scorpions, though, Eurypterids are predators eating fish and other marine organisms, some growing over 6 feet long, such as Jacalopterus. They were clearly well adapted to this niche, as they survived over 200 million years from the early Ordovician to the Permian Triassic extinction. Eurypterids likely explored the land long before our tetrapod ancestors ever left the seas. And indeed, a Carboniferous Eurypterid, Hibbertopterus, currently holds the record for the largest terrestrial arthropod trackway. And we reach Arachnida, chelicerates with 8 legs. This class is quite diverse, with members ranging in size from microscopic mites to tailless whip scorpions whose leg span can get over 2 feet, though Gigantoscorpio in the Carboniferous got up to 3 feet. This class originated in either the Cambrian or Ordovician and subsequently terrestrialized in the Silurian. Arachnid book lungs look like internalized gills. Likely the most well-known group of the arachnids, the spiders, Aranea, have a fossil record going back to the Devonian, such as Attercopus. However, the earliest spiders sported long tails and lacked spinnerets. Chimerachne, preserved in Cretaceous amber, shows that these strange spiders persisted alongside more modern-looking spiders for many millions of years. The other great branch of the arthropod tree is Mandibulata, so called for their unique mandibles, including the myriapods, crustaceans, and hexapods. Myriapoda contains the carnivorous and often venomous centipedes, herbivorous millipedes, and the small blind symphylans and pauropods. Though myriapods are related to insects, fossils linking the clades have been lacking. Of course, the fossil genie came to the rescue, plopping Heterocrania in early Devonian Rhynie shirt, possessing characteristics diagnostic of insects and myriapods. And yes, the most famous fossil member of the clade is likely the Carboniferous Arthropleura, which measured 8.2 feet. There has been some debate as to when myriapods, representing the earliest terrestrial arthropods, made landfall. Molecular clock studies estimate that myriapods terrestrialized as early as 500 million years ago, the late Cambrian, synchronously with or immediately after terrestrial plants. However, the earliest fossil evidence of terrestrial myriapods, an extinct group closely related to millipedes known as Campicaridae, comes from 425 million years ago, the late Silurian, which was long after plants colonized the land. Deposits in Scotland show a radiation of myriapods where only a few are terrestrial about 425 million years ago. By 420 million years ago, Campicarids and centipedes, as well as arachnids and eurypterids, had invaded the land, and by 414 million years ago, myriapods had become diverse. If there were any terrestrial arthropods prior to 425 million years ago, they were likely small, and possibly lived in the sediment, obscuring their fossils. As for crustaceans, their monophyly has been shredded by molecular studies. They were previously claimed to comprise a single clade called Crustacea that was sister to Hexapoda. However, it is now understood that some crustaceans are more closely related to the hexapods than to other crustaceans. For example, ostracods are sister to the rest of the crustaceans, while Malacostraca, the crabs, shrimp, and lobsters, as well as copepods and barnacles, group together. Barnacles will get their own future tale. And Branchiopoda, the fairy shrimp, and Remipedia, the small blind crustaceans with many repeating segments, are very close to the hexapods, with researchers suggesting that the split between these crustacean clades and hexapods occurred in a shallow marine environment. That brings us, at long last, to today's tale, the upside-down brine shrimp. Branchiopoda is a class of crustaceans commonly referred to as fairy shrimp, and this class encompasses 9 orders, containing such members as Triops, which are sold as pets, Daphnia, the water fleas, and clam shrimp, which look superficially similar to those bivalved arthropods from the Cambrian. The order containing brine shrimp is Anastraca, and all 300 species within it swim upside down. Just like the previous tale, we're going to depart a bit from Dawkins and Wong's telling. The point of the tale is that, according to Dawkins and Wong, brine shrimp and a few other animals we'll mention are recent reenactments of a process that occurred some 540 million years ago. Remember from the Lancelet's tale that chordates experienced a dorsoventral inversion, where the genes that regulate the development of our dorsal side regulate the ventral side of protostomes and vice versa. Dawkins and Wong discuss that brine shrimp are also inverted. The dorsal side of brine shrimp faces the sediment, while their ventral side faces the water's surface. Perhaps, Dawkins and Wong surmise, brine shrimp inverted geologically recently, and 100 million years from now, their traditional dorsal side will become their new ventral side. Furthermore, the authors posit that the mechanism for the inverted behavior of brine shrimp may be due to the Baldwin effect. In essence, this is the idea that when an animal becomes exposed to some new environmental pressure, some of their behaviors may change as a result. Genetic variations that encode these behavioral changes are therefore more likely to be positively selected, and thus, the former purely behavioral changes become genetically encoded within the population. Dawkins and Wong speculate that maybe the Baldwin effect played a role in our distant chordate ancestor. For some reason, chordates began swimming upside down, and their dorsal and ventral sides switched, resulting in our modern condition. So, is this scenario for brine shrimp evolution, and more broadly, the evolution of chordates, correct? Well, not exactly. Brine shrimp did not invert geologically recently because, remember, the whole of Anastraca is inverted, and their common ancestor is estimated to have lived some 430 million years ago in the Silurian period. There is even a late Cambrian stem Anastracan called Rebbachiella that also may have been inverted. Thus, the suggestion that Anastracans will completely invert their body given 100 million years appears to be false. They have been swimming upside down for 4 times that long. This raises the question, why do brine shrimp swim inverted? For these tiny shrimp, feeding and swimming are inextricably linked. On each of the 11 or more pairs of swimming limbs, the brine shrimp bears projections that filter food particles such as organic detritus, algae, ciliates, and rotifers out of the water. Brine shrimp swim in the horizontal plane, so swimming inverted brings the center of gravity down, stabilizing the shrimp, which is important for swimming in open water, as well as slightly reducing the energy needed for maintaining balance. However, this method of swimming is only beneficial if the animal possesses a large number of identical limbs. Swimming upside down wouldn't be useful for a lobster, for instance, who has chelipeds, walking limbs, and pleopods. But this issue can be circumvented if a crustacean with different types of limbs encloses those limbs in a bivalve shell and only swims with the cephalic appendages. Inverted swimming is also very useful in the environments Anastracans tend to inhabit. Most Anastracans live in ephemeral, shallow freshwater pools whose substrate is covered by a layer of loose detritus and fine inorganic particles that are easily stirred up. An individual swimming in the traditional crustacean manner near the bottom of this pool is inevitably going to take unwanted particles into the food groove. Thus, swimming inverted allows brine shrimp to capture organic particles without becoming clogged by unwanted ones. A number of other crustaceans also swim or sit upside down. The Daphnia genera Cymocephalus, Scapholebris, and Megaphenestra, the Chidorid Dedea macrops, the freshwater ostracod genera Notodromus, Nunhemia, and Onchocyprus, and barnacles, subclass Cirripedia. Outside the crustaceans, the insect family Nodonectidae is commonly called backswimmers because they swim upside down just below the water's surface, and the Cambrian bivalved arthropod Fibulacaris nereidus has been interpreted as an upside-down swimmer based on its backward-pointing eyes and 40 pairs of identical flap-like legs similar to those of brine shrimp. Vertebrata has its own inverted swimmers, the upside-down pipefish Heraldia nocturna, and the upside-down catfish genus Cynodontus. Cynodontus are found in East African rift lakes like Lake Victoria and Lake Tanganyika, famous for the radiation of cichlids we met in the cichlid's tail. Cynodontis inverted swimming is related to both catching insects and their facultative air breathing. Evidently, drag from surface waves is reduced by their inversion. In the ribbonworm phylum Nemertea, both Baleonemertes and Cephalothrix suni from Vietnam evidently crawl on their backs. And as for mollusks, the sea slug Glaucus and giant clams of the genus Tridacna swim inverted. A common theme for all these animals is that the side facing up while swimming is always more darkly colored than its opposite side. The upside-down catfish Cynodontis nigroventris is named specifically for this feature. Its belly is darker than its back. This is called reverse countershading. Typically, countershading involves having a dark back and a light belly, because the dark back camouflages the animal against the seafloor, while the light belly camouflages the animal against the water's surface. Reverse countershading is the same concept, but the colors are flipped because the animal's surfaces are flipped. Two final animals deserve an honorable mention even though they aren't upside-down swimmers. The first is the cnidarian Cassiopeia, which is commonly known as the upside-down jellyfish. This jellyfish has a symbiotic relationship with the dinoflagellate Symbiodinium, just like coral, and even requires the dinoflagellate to complete its metamorphosis. Cassiopeia is unique among jellyfish in having its flat exumbrella pushed against the seafloor while its tentacles face upwards, sort of like it's impersonating an anemone. However, jellyfish don't have a dorsal ventral axis, so they aren't really upside down. The second honorable mention is the razorfish Aeoliscus punctatus, who doesn't swim upside down, but vertically. This is an adaptation to hide among urchin spines and is facilitated by the fish's center of buoyancy, also nearly being its center of mass. So that's the corrected brine shrimp's tail. The ancestor of chordates inverted in the early Cambrian, and various clades of animals have also inverted in the years since due to different selective pressures. So thanks for watching, and we'll see you all next time.